imagine sort of every neuron is an axis, right? So if I have a million neurons, then I have a million dimensional space. So I have a million axes, right? So those trajectories in that million dimensional space live actually in a much lower dimensional subspace in there. That's the secret sauce that allows us to have a hope of understanding cognition, but only recording less than 1%. If you were, an engineer and you went to your boss with a model of a biological brain, you just get fired. It's just the neurons are just really bad at responding to things. Yet the brain does amazing things. It's able to do inference at levels that sort of are beyond what machines can do often.
So what exactly is computational neuroscience? It combines the two, but how so? Have you ever wondered how billions of neurons somehow work together to create thoughts, memories, decisions, and even consciousness in cells? Why does the brain sometimes behave in predictable patterns, while at other times it seems almost chaotic? How can math and computation help us understand something as complex as the human mind? These questions sit at the center of one of the fastest growing areas in neuroscience, computational neurodynamics. From the synchronized brain activity to neural networks, from decision-making to disorders of the brain, scientists are beginning to uncover the hidden rules that govern how neurons interact over time, and how these interactions give rise to behavior, cognition, and learning. Hello, I'm Avnish, and this is The Early Perspective, a podcast where we explore how the brain learns, how it works, and how it changes over time, especially what that means for how we learn, think, and live. Today we're going to be diving into one of the most fascinating topics in neuroscience today, computational work and neural dynamics, an area of research that combines neuroscience, mathematics, physics, and even computer science to better understand how the brain functions as a complex system. When most people think about neuroscience, they picture brain scans or individual neurons firing. But computational neuroscience asks something deeper. How does enormous networks of neurons coordinate activity? How does the brain process information so efficiently? And can understanding these dynamics help us better understand intelligence, behavior, and neurological disease? I'm incredibly excited to welcome today's guest, Dr. Brent Doiron. Dr. Doiron is a professor of neurobiology and statistics at the University of Chicago, where he also serves as the chair of the Committee on Computational Neuroscience. He originally studied physics and mathematics at the University of Ottawa before earning a PhD in physics. He then transitioned into theoretical neuroscience during his postdoc fellowship at the New York University. Over the course of his career, from the University of Pittsburgh to the University of Chicago, his research is focused on understanding how large populations of neurons interact dynamically to process information in the brain. His work combines neuroscience, mathematics, computation, and statistics to study topics like neural correlations, cortical networks, variability in brain activity, and how collective neural dynamics shape information processing. I'm really excited for this conversation to explore how neural dynamics can help us better understand the brain itself. Dr. Doiron, thank you so much for joining me today.
Thanks so much for having me. This is exciting. I love reaching out to sort of younger audiences. Thanks for giving me the opportunity.
Yeah, of course. Okay, so before we get straight into the conversation, I just want to go back to the beginning. Could you tell me a little bit more about your journey into neuroscience and computational research?
Sure. I started actually even as an undergrad at 98, when at the University of Ottawa, I got put on a project that the professor I was working with said was neuroscience. I had no idea about the brain, nothing like that. Neuroscience at the computational side was sort of in its infancy at that time. So I started there, and then I sort of got hooked. And then I absolutely did a PhD with that same professor at the University of Ottawa. My PhD was in physics, but I worked looking at neural computation from a physics point of view, specifically working on an area of neuroscience called neuroethology, which is where they look at specialized organisms. They look at neural computations that certain organisms have specialized. And the organism I was working on was the weakly electric fish. It's a classic neural prep where there are fish that produce electric fields that surround their body. And then when objects that have a different impedance than the water around the fish come in, then the field lines will morph. And then on the surface of the skin of these animals, there's receptors that are able to see that. It's almost like having 3D vision, but it's in a fog because the electric fields decay quickly. The reason I think people studied this in the nervous system is there's sort of lots of control for this. We know how to manipulate electric fields. We can also these animals produce electric fields when they're paralyzed, so you could study them in the sort of 80s and 90s when people weren't really doing awake behaving, especially in fish. It's very difficult to do that. Now people are doing that. Anyway, that's how I started working on sort of mathematical modeling of that. And then during my postdoc, as you mentioned, then I sort of walked into sort of the broader neuroscience community at NYU, and then found myself in mathematics departments through most of my career. But then now here I'm in a bona fide neuroscience department here and statistics. But I've been slowly over the years becoming more and more of a neuroscientist. Now that's how I think of myself.
Yeah. Was there any specific moment that really pushed you into the field of neuroscience particularly, or was it just that project?
Well, I was interested in the 90s. People were really getting hot about complex systems in general. They've changed over the years, but in the 80s and 90s, there was a lot of excitement about that. And one of the areas that I wanted to work on was actually social insect dynamics. So I wanted to work on like how bees work as communities or ants work as communities. But I didn't have an option at University of Ottawa. And then sort of that's how I got convinced to say, well, why don't you try looking at neurons? Neurons are, simple-ish agents. They're not actually that simple, but they work as a collective. And the mathematics, the kinds of mathematical techniques we use to study groups of neurons are similar to how we study sort of groups of different simple agents. And then that pushed me in the field, and then I got hooked, and there was a lot of excitement about comp neuro in the sort of early 2000s, and there continues to be today. So I felt fortunate to sort of jump on a bandwagon early.
Yeah, I think it's very interesting that math and neuroscience really work together and complement each other in such a interesting way. I think a lot of people don't realize that they are so similar, and they think that they're separate worlds. But I know we briefly touched on this, but what exactly is computational neuroscience? It combines the two, but how so?
Yeah, I think there's computational neuroscience is a broad term, and it probably differs depending on who you're talking to. I think sort of there's two branches of this. One aspect of computational neuroscience is building new analysis tools, given just this vast amount of data that just every year, the amount of the brain that we can measure at one moment in time and the scales that we can measure that brain activity at, change all the time. And we need to have quantitative tools to be able to catalog and analyze that data. And now the fact that people are really doing connectomes, where they're basically going in and measuring every possible connection in right now fly brains and worms they work on. Those are massive data sets, so we need to have ways to distill or extract the salient aspects of these data sets. Mathematics is great at doing that, and sort of machine learning, which is sort of a branch of statistics here, really pushes that. That's one branch of computational neuroscience, helping people organize and interpret data. The other branch that I'm more invested in myself in my research group is modeling the nervous system. We know a lot about how single neurons act. We know how to model single neurons. Actually, there's a Nobel Prize for this in the 60s from Hodgkin-Huxley. So having electrical circuit models of neurons has a long tradition. So we know how single neurons work, and we know how synapses, the thing that connect neurons together, work. Then it makes sense to start expanding our models to have groups of neurons that interact with one another. So these are more mathematical models of brain circuits, much more in the tradition of statistical physics, where we sort of have simplified, I don't want to say spherical cow, but oversimplified models. And they're trying to ask, what are the critical aspects of the neural activity or the connections between neurons that are needed to explain brain-wide neural activity or even behavior. So those are sort of circuit biological models. That's more the side I'm on. Of course, there's interactions between the two. We need to analyze big data sets to validate our models. So it's really silly to say these are separate areas, but there's a spectrum.
Mm-hmm. So then, I know that you talked about Hodgkin and Huxley and that model, but then what exactly are these models telling us which are not, I mean, at least which experiments can't say alone?
Well, so Hodgkin and Huxley, they were actually physiologists. They were sort of they was really one of the earliest sort of gold standard models of neurons came from two physiologists that were very mathematically trained. So the Hodgkin-Huxley model talks about how different ionic species, in particular potassium and sodium, how potassium and sodium interact with one another in terms of how they flow across a membrane in order to produce the action potential, which is sort of the central currency of information in the brain. People call them spikes. So neurons produce these spikes, and Hodgkin and Huxley really figured out how these spikes happen. And it was really about an interaction between two different ionic species. Experimentally, you can isolate one species from another, but if you want to sort of understand how they both go together, that's experimentally very difficult to do. So what they did is they played this game where they would isolate these from one another, build a mathematical framework that when put together should produce an action potential. And then lo and behold, it did produce an action potential. The cool thing about what it was sort of a complicated set of equations that now, of course, if you wanted to numerically solve these on your phone, it would be trivial. It would be, milliseconds on your phone. But back then, Hodgkin and Huxley, they were doing these calculations in the late 40s, where if you wanted to do anything with a computer, you had to be working with the military, the Manhattan Project. Like there was no computers to really do these things. Yeah. So Huxley, he was the student of the pair. He ended up doing this with a crank calculator. So he had like an accountant's calculator with and he did the numerics. So that took him weeks to do calculations that we would do now quickly. So these guys were super ahead of their time. But to answer your question about what this gains, we now know how different ionic species interact with one another to produce neural activity, and that sort of gave the birth of pharmacology because we could manipulate the level of these ionic components with different drugs. We could block components, we could enhance components, and sort of the birthplace of sort of pharmacology at the cellular and synaptic level was sort of at that time. And that, of course, has a history today. Pharmacology and mental health have a big component. So I wouldn't necessarily say it all started with Hodgkin-Huxley, but understanding the biology of neurons and how chemicals can affect that biology started with.
Right. So then you're saying that especially these models can provide us more insight that experiments alone can't just allow us to find, right?
That's right. So in today's world, it's cheap to do numerical analysis, much cheaper and easier than it is to do experimental work here. So you can use these models if they're well made and they're well constructed and well validated, then you can use them as a hypothesis generator for future experiments in a way that, now you can do many, many, many numerical experiments cheaply and quickly. And then the most performing ones in the sense of manipulating neural activity might be the ones that then you would first try to do experiments to replicate.
But now with the advent of better technology, calculators, and even better AI and machine learning, are these models going more obsolete because we don't need at least the computational, at least the literal calculations which these models provided, or at least let us, I guess, do it faster, right?
I think interpretability. I certainly think I can. When we look at, for instance, the Hodgkin-Huxley model, I can understand that solution in terms of ionic flow, in terms of sodium, potassium, in terms of nonlinearities, in terms of how channels that are holes in the membrane, how they act. This is a super interpretable, well-founded theory. If we're going to start throwing AI and not asking AI to be interpretable, then sure, you might be able to predict things, but having an understanding of what's happening might be out the window. I think there has to be a good balance between these two. So, the classic modeling that, physicists and computational neuroscience at a later point do gives us a simple interpretability that we can talk about our models with one another. Whereas if it's just straight AI with no sense of how things are happening, it's not as exciting to some of us.
Right. The models are providing a level of understanding that AI alone can't probably do yet, at least.
Not yet, and it'll be a back and forth between. AI is of course making huge advances in pure mathematics because those problems are much more crystallized, right? Where there's very well established frameworks. So pure mathematics is going through a bit of a renaissance right now because of that. I don't know if we're there yet in applied mathematics or applied physics or mathematical biology is not quite there yet. Who knows what the future holds? But definitely saying you're not going to use AI in your research program is sort of foolish. I think a lot of us are adapting to using AI as a tool or as a blackboard to work off of. Asking it to do everything and then being unaware of what happens is probably not the way to go. But saying you're not going to use AI to study new problems is probably limiting yourself.
Yeah. I know another thing that you study is neural dynamics, and that is one of the main topics of our episode. But, of course, this is a topic that most people would not even be aware of. What exactly does it mean when you say neural dynamics? What is a scientist talking about?
Sure. So, of course, all of sort of cognition and responses and sensory inputs and movement, everything happens dynamically in time, right? So the fact that neurons unfold and their activity unfolds in time is nothing special. What people talk about when they say neural dynamics that makes it interesting is that a lot of the movements that we make or the decisions that we make or memories that we store for short term or sensory inputs that we respond to, their dimension, the effective number of variables of these is far less than the number of neurons that are actually enacting these computations. So this movement is much lower. There's millions, tens of millions of neurons in the motor cortex that are controlling movement, but yet I could describe my movement, in a much lower dimensional space. 20 dimensions. Joints that move in certain directions. So that's true for movement. For sensory inputs, the salient aspects of sensory inputs can be low dimensional for tasks we want to do. Decision making is in principle at its most primitive sense is just a binary variable. Yes, left, right. No, yes.
Yeah.
So these are very, very low dimensional things. When people talk about neural dynamics, they want to say how do we go from the high dimensional space of neural activity to the appropriate low dimensional space that governs movement, cognition, sensory representation. So when people say neural dynamics, they're really thinking about what's the low dimensional space in which neural activity and its dynamics should be thought of unfolding in. Yeah. So a lot of this deals with dimensionality reduction. That's an area of machine learning,
right?
Which ultimately, if people are interested in comp neuro, one of the best things you can do is learn linear algebra.
Yeah.
So linear algebra tells us how to think about spaces and how to think about subspaces in those spaces. And that's exactly what neural dynamics does. What's the appropriate subspace that the important activity of the nervous system is happening in?
Right. Then if it's so multidimensional, how are scientists able to reduce it at least into something interpretable at least in what you were talking about? Yeah.
Sure. So if you can imagine sort of every neuron is an axis, right? So if I have a million neurons, then I have a million dimensional space. So I have a million axes, right? And then at any moment in time, the activity of those million neurons is a vector in that space or a dot in that space. And then as activity changes, you get a trajectory. And that trajectory in principle lives in this super high dimensional space. But when you ask, is this neural trajectory, because neurons are interacting with one another, one neuron might be competitive with another neuron in the sense that when one neuron's active, it will suppress the activity of another neuron. So given that competition between the two, they're tethered to one another. So they're not independent neurons. When one's up, the other one's down. When one's down, the other one's up. So all of these interactions that are happening through the circuit in the nervous system is going to put all these constraints. So those trajectories in that million dimensional space live actually in a much lower dimensional subspace in there. And identifying that subspace and how that activity goes in that subspace is how we can interpret in a reasonable way how neural activity subtends decisions, which are low dimensional, movements, which are low dimensional, representation, which is often low dimensional. Identifying that space is the first step at sort of asking, how is this collection of neurons really doing the job as a group?
Right. So then these lower dimensions are just essentially interpretations of the relationships between the different neurons, and understanding that can help you reduce it in a way.
That's yet from a mechanistic point of view, I definitely think that both how inputs drive a bunch of neurons as well as how their interactions between each other, those are signatures of that low dimensional space, or that low dimensional space shows signatures of an activity, I should say. It's, both sides of the same coin, but also how it's much easier for some downstream center, some area that this group of neurons is projecting to. It's much easier for that downstream center to interpret low dimensional activity. So to build decoders, often we build decoders of the nervous system, but one area of the brain has to decode what's happening in another area of the brain, right? That's how the brain works. And having that sort of having a low dimensional representation in one area makes it so much easier for another brain area to help understand what's happening there and to build a neural code for whatever aspect of neural activity we want to talk about.
Can these patterns of neural activity tell us anything about behavior or cognition?
Sure, yeah. Well, certainly one classic framework is if we have decision making, right? And an animal is integrating some evidence, is trying to decide. I mean, often these tasks are some noisy signal, and the animal has to sort of detect what the signal is in this corrupted framework. So it takes some time. So they have to integrate evidence. And then at some point in time, they'll make a decision, yes or no, left, right, whatever it is, right? So if you think in a low dimensional framework, basically what you have is sort of two, what people call attractors. If there's a dynamical landscape there, then there's a point in that space that is the yes decision, and there's a point in that space that's the no decision, and there's a point in that space that separates the activity that will go yes or go no. That can all happen effectively. I've just described something that's a one-dimensional thing. It's a line, right? Maybe the yes is at that yes is at 1, no is at -1, and the decision boundary is at 0, right? So I've collapsed decision making that happens in a very, very high neural dimensional space onto just a one-dimensional line here.
Right.
And that's all you need to think about binary decision.
Yeah, I think it's really fascinating, especially this allows us to interpret exactly what's going on in the brain in a way that we can genuinely understand, despite it being so complex and so multidimensional, as you were talking about.
Exactly. It's how do you extract the salient aspects of neural activity to help you understand the thing you want, which is often cognition, right? So how do we do that? When tasks that we get animals to do, or even humans to do, end up being much lower dimensional than neural activity, then that it's really useful to try to understand neural activity in the dimension that's appropriate for the cognition, not living in this high dimensional space that's just a spaghetti of neural activity. I don't know what to do with that. But if I can collapse it to a line, and I know that points on this line mean things like yes or no, or maybe, then great. Then I've learned a lot. And if I can do that rigorously through rigorous mathematics, then I'm confident that this is a reasonable path to take.
Yeah, especially because if you have a lot of data points going all over the place. You can't really understand anything without it genuinely being reduced in a way
That's right.
that you can see and visualize. Yeah,
Absolutely.
for
I'm a
sure.
simple guy. I need things to be simple.
Yeah. And then just to pivot, since we did talk about AI and artificial intelligence in general, how are biological neural networks similar to artificial neural networks?
Well, so I mean, one in part, AI in its infancy really started from thinking about binary neural agents. Actually, some of it happened here at U Chicago with Pitts, McCulloch and Pitts. They were two researchers that were sort of really influenced by Neumann about how to think about sort of the first model of the nervous system, which is binary agents, right? And these were the earliest stories of neural networks. But so how are they similar is that it's also this very high dimensional space. There's lots of agents in an ANN, artificial neural network. There's lots of agents. They interact with often complex wirings, so often random matrices, or if it's a learned system, as they often do, it's still a pretty complicated interaction matrix that's sort of devoid of symmetry. And symmetry is one thing that physicists use all the time. And sometimes, neural networks, ANNs, aren't as symmetric as we'd like to be able to, take whatever statistical mechanics tool we have in our toolbox and not go apply there, right? So that's in one way how ANNs are similar to actual brains, is heterogeneity and complexity of the interactions. One way they're really different is that the nervous system, both individual neurons and synapses that link neurons, are very, very unreliable. There's a lot of noise in the nervous system. Synapses, if you were an engineer and you went to your boss with a model of a biological brain, you just get fired. It just the neurons are just really bad at responding to things. Sometimes they respond, sometimes they don't.
Not
Sometimes they
reliable.
respond a lot.
Yeah.
Synapses fail all the time. The warranty on these things is really low. So, but yet the brain does amazing things. The brain does, it's able to do inference at levels that sort of are beyond what machines can do often. So how does it work with how do biological brains do such amazing feats of cognition with such, poor parts, is a super interesting question, right? And maybe the variability we have there can be used to do computation, or maybe because computation is done in these low dimensional spaces, they can organize these spaces to be orthogonal or perpendicular to noise axes. So if you have a lot of noise that goes in one dimension of the system, the best way you can do computation is often make your computation to be perpendicular to that axis of noise. And then who cares if it's noisy? It's not affecting the low dimensional computation.
Yeah.
Yeah.
Is that one of the biggest questions I mean, at least that scientists are trying to answer right now? Or have we come close to at least answering in what way the brains has so much prowess compared to AI, at least, in handling these, as you put it, poor quality parts and getting better results?
Yeah, it's a great question. It's often task dependent. I mean, certainly ANNs can do things that better than we can do, right? So there's certain kinds of computations, and that's why we use them. But it's an open question. I think it's a little ill posed in the sense that the answer to that question, which is sort of general, is probably going to be dependent on the task, the animal, the brain area
Right.
that we talk about. There are certain areas of the nervous system where reliability is premium. So they certainly do this. So there's certain areas where that the variability I was talking about is something that is minimized. In cortex and hippocampus, the variability is present. Some of that variability may be the fact that on every trial, when an animal decides to do something is different on every trial. So if you can organize and compare your trials against the right time axis, maybe some of that variability will go away. People have looked at that, and that's in part true. So part of it may be explaining the variability I was talking about. Maybe it's not completely due to the fact that synapses and neurons are sort of unreliable. They're unreliable, but maybe some of this is our inability as researchers to sort of control all of the nuisance variables that are out there that make individual trials different. So as we get better at understanding how to probe the nervous system and to control unwanted aspects of variability, then we can hone in on what the true variability of the nervous system is. That's probably where some people are putting their efforts now.
And is reducing this, at least the unknown and these other extra variables, still similar to just reducing the dimensions, or is it just a completely different process?
That's what that's doing, is sort of reducing the uncontrolled variance. So, for instance, I'll give you a concrete example. When you're looking at activity of neurons in the visual system, right? So there you're recording in V1 or V2. These are area of the visual system's a pathway. When you're recording, let's just say V1, you'll see that neurons on a trial-to-trial basis, even if you give the exact same stimulus, on a trial-to-trial basis, they will fire different numbers of action potentials and at different times, right? But you can reduce that variance significantly if you train the animal to focus its eye at the middle of the screen. If on every trial you let the animal or human to saccade over the image, and that saccade pathway is different on every trial, even though the image is the same, the sampling of that image is different in every trial. So that variability you see in the nervous system isn't due to neurons and synapses. It's due to the way the animal's sampling that image. So if you can say, no, don't move your eyes, focus, you can do that. You can train animals to just look at a spot. And then a lot of that variability I was just talking about goes away, but not all of it. There's a lot of variability that remains. So, and then when you can sort of get rid of all of the sort of behavioral uncontrolled variability that we can, then sort of looking at the residual variability that might be neural is that dissection is something that people are looking at.
Right. So there's just a lot of variables, a lot of factors that just go into
behavior is super variable, and especially when you're doing it with monkeys or mice in particular. You cannot really train a mouse to stare at a stare. You can train monkeys, but you can't train a mouse to do it. Mice are just going to look where they're going to look, and that's it. You can't. What are you going to do? And flies are going to be even worse. Yeah.
And then, of course, in many ways, actually, the human brain happens to be even more efficient than AI because of a lot of what we talked about. So when we're studying the human brain, why is it important to study groups of neurons versus just a singular neuron itself?
That's a really good question. I think the earlier stuff in the 90s from Stanford professor Bill Newsom and Michael Shadlin and Tony Mofshin and Bill Newsom's graduate student Ken Britton, they looked at trying to decide what the motion of a visual field was by recording from neurons in an area of the brain that is sensitive to motion here. And they were able to sort of decode the motion of activity just by sort of looking at a single neuron. So they were and to predict the animal's decision. These animals were doing a task that was, what to decide whether motion was going one way or another way. Here, this again, one of these noisy signals. So it's not so straightforward on how to do it. And they were able to sort of mimic the decision making of the animal by just looking at a single neuron. So back in those days, a single neuron was enough. Now the task was low dimensional, and probably the reason they could do that is that single neuron was getting inputs from a bunch of other neurons. So you're just looking at a readout of somebody that's looking at lots of neurons. So it's not that single neuron was doing all the work. It's that it was working with its buddy neurons, and we could read out that group collective activity from one neuron's behavior here. So when we started recording from more than one neuron, then we could start understanding more complicated tasks that you couldn't really extract from just binary single neurons. So movement in particular, I think trying to understand how movement works, that really sort of the Krishnan Shenoy, who was also another Stanford professor, him and his sort of legacy of postdocs and graduate students that are now faculty at amazing places, they started to start thinking about how collective groups of neurons could control complex movements, which couldn't really be understood from just a single neuron there. So really looking at, so I think even when you look at a single neuron, you're looking at how the collective group of neurons are acting, and you're just reading it out from one neuron. So it's never the case,
Right.
other than in super simple situations where a single neuron does all the job.
Mm-hmm. So then, especially in cases where you're involved with higher dimensional things, you need to study a group of neurons to at least try to understand and analyze exactly what's going on, because a single neuron doesn't have enough information.
When you had very, very simple tasks, you could project that group activity through a single neuron's output and understand things. But the more complicated the tasks are, you're not going to be able to look at a single neuron. We're always looking at subgroups of neurons. I mean, even though there's millions and millions of neurons, the best labs now can record 1,000 neurons at once, maybe. So still orders of magnitude smaller numbers of neurons that are engaged. And we can really reconstruct animal behavior and animal decisions quite faithfully when you have 1,000 neurons. So
right.
That's super fortunate. I think neuroscience would have been, dead in its tracks if you had to record from 90% of the neurons in the brain to have a hope of understanding what the brain is doing. Then we'd never do anything. Yeah. So but the fact that you can get a sense of what the nervous system is doing by looking at a small group of neurons, because that group of neurons is talking to the guys you're not looking at and reading them out. And then now we can read out their activity. And that activity is the low dimensional framework. That's the secret sauce that allows us to have a hope of understanding cognition, but only recording less than 1% of the neurons available. Yeah.
Are all activities and behaviors a result of neurons interacting together, or is there anything which is just a single neuron performing a task, for example?
Well, in simple organisms like escape movements in fish, there's this like Mothner cell that is a single neuron, and it's if it's driven, it'll get an escape reflex in an animal. That's an example where a single neuron will be important. So in invertebrate systems, there might be examples like that, but I think sort of, how does a neuron know what's going on? If you have a neuron that's controlling movement, even in the simplest organism, really, other than the Mothner system, if you have a single cell that controls movement, it's getting input from a sensory neuron that's looking at the sensory world. So you really need like at least two neurons there, one to look at the world outside and one to move muscles in response to that world, right? So you really do need, in that scenario, two cells, and lots of invertebrates, worms, flies, all that stuff. They have a collection of neurons much smaller than we have here, and it's much more stereotyped. But having just a bunch of neurons that don't interact with one another, the only time I can say that that's probably the case, I guess, is in the cochlea, one area. So the way we understand sound is we have a bunch of cells in our sort of subcortical areas in the inner ear, the cochlea, there, where individual neurons there are tuned to specific frequencies of sound. So an acoustic sound has a rich component of frequencies. And individual neurons in an array of neurons, or what people call a line of neurons, a lateral line here, then when individual sounds come in, low frequency sounds will make one neuron fire, high frequency sounds will make another neuron fire. Those neurons don't talk to each other. But in order to understand the complex acoustic sound, somebody has to look at all of them. Yeah. So the neurons don't talk, but as a group, they are responsible for coding a part of the sensory scene. And then as a collective, somebody looking at them all can get a sense of the whole sensory scene.
Mm-hmm. Yeah, especially, I guess most people would not realize that any simple action involves so many neurons and so many different systems it's not just, of course, the processing. You need to process the visual information, and then you need to actually take action, the muscles, and there's so many different parts of that system that people just fail to account for most times.
Yeah, and I think a lot of what we've thought about in the nervous system is that these jobs are compartmentalized. There's parts of the nervous system that's responsible for vision. There's parts responsible for auditory responses, somatosensory responses, movement, decision making. So we think that there is, this compartmentalization of neural function. That idea has been challenged a little bit. That's a fun thing in the last sort of 10 years. When you start recording from mice in particular, you'll see activity in visual areas of mice that really are connected to the motor movements of these animals here. So probably what's happening, or we think what's happening, is motor signals in motor areas don't just project to muscles, to move muscles, but they project to sensory areas to tell that sensory area, hey, we're moving, right? And maybe you should change the way that you're interpreting this visual input. If a mouse, for instance, is stationary versus running, the visual field is very different. When you're running, objects are coming at you all the time really quickly. When you're stationary, and you're not afraid of that. When you're running and objects are coming, you just have to move out of the way of a stationary object. But if you're sitting still and objects coming at you, you get scared because something's moving towards you, right? So there has to be a change of interpretation or context depending on whether you're running or whether you're just passively experiencing the world. And it's maybe reasonable that motor signals might go down and influence sensory areas, visual areas for that. So, but you tend to see then that computation isn't just happening isolated in one area. It makes its decision, goes to another area. That area makes its decision. There's this interconnectedness between different brain areas. And looking at a complex task will probably have representation in lots of different brain areas at once. And sort of understanding how we should tweeze those representations apart is sort of a new area, because we can look at more brain regions at once than we could have sort of 20 years ago.
Right, yeah. Almost all the parts of the brain are working together once, and they're all like systems which are interconnected, right?
Yep. So it's where do we live? We have an interconnected system, but yet components in that system do have different computational roles. We don't want to think of the brain as just like a soup where all neurons are all over the place and it doesn't matter, they're all the same, right? So that's how this differs a lot from statistical mechanics, right? Where there you often have very similar particles interacting with one another. So in the nervous system, how different neurons in different brain areas wire together, what are their biological features, these are all sort of differences. So we think there's compartmentalization or specialization of activity in certain brain areas, but they have to interact with one another in order to perform interesting computations. So how do you navigate, that compartmentalization and specialization with the interconnectedness required to do, real interesting computations? That's a challenge.
Yeah, for sure. That's really fascinating. And I also want to pivot into another major question in neuroscience, of course, that is disorders of the brain. How can computational neuroscience help us better understand these neurological or psychiatric disorders?
So that's a great question. And, there's an area of psychiatry called computational psychiatry. That's been around for about 15 years now. There's pushes there. I will say a lot of disease states, a sort of a zeroth order model. Zeroth order model means a model that's like the base level model that clearly needs to be built upon. But a lot of neurological disorders that we see come from an imbalance between the amount of excitation, the amount of excitement that neurons have interacting with one another, from the amount of inhibition, so the ability for one neuron to stop another neuron's activity. And we see that a lot of disease states can be characterized, not understood necessarily, but diagnosed in part with an imbalance between excitation and inhibition. Epilepsy, for instance, is sort of a lack of inhibition. So when you have lots of neurons that just want to excite each other, then everybody gets excited, all the neurons fire, and that's a seizure. That's what a seizure is. When you have uncontrolled, massive activity of a bunch of neurons, then you're in a seizure. So inhibition, or the ability for neurons to stop one another, is super important. And when that imbalance happens, we see similar things in schizophrenia. We see even some signatures of excitation and inhibition balance in autism. Of course, the challenge is epilepsy and schizophrenia and autism aren't even remotely the same at the behavioral level, right?
Of course.
So there is a signature of those diseases that are the same, excitation and inhibition, but how exactly that imbalance manifests itself in sort of the phenotypes, the behavioral phenotypes of these diseases, is an open question. But computational neuroscience can help us understand what it means for excitation and inhibition through models of the nervous system. How when you dysregulate that excitation and inhibition, what happens, right? And then maybe how can you recover that if you have an abnormal circuit? What's the best way to push it into a regime where it acts like a normal circuit?
Yeah, for sure. I think that's a really interesting way to think about it, because a lot of these disorders are related to the inhibition and excitation, as you're saying, especially the seizures. So are these disorders also connected to abnormal neural dynamics?
Sure. So if you have, often what can happen is if you have sort of unregulated interactions amongst excitatory cells that aren't stopped by inhibition, what sometimes will happen there is the dynamics of the system can become very low dimensional. A seizure is really like a one dimensional, everybody goes up, everybody goes down. It's this huge thing. So that's a super low dimensional dynamic. So it's possible that in that framework, you don't have the necessary space to be able to do interesting computations, because it's been hijacked by this ultra low dimensional dynamic that is not in the right direction to do computation. So that's a possibility there. That's a very simplistic model for that. But yeah, if you by identifying the right low dimensional spaces that cognition should operate in, and then asking when we have an abnormal circuit, perhaps because there's too much interaction that's putting too much coordination among cells, how is that destroying our computational model of a cognitive brain working properly in this low dimensional space?
Right. Yeah. Definitely, I totally get that. Okay, so before we wrap up, I just wanted to do a quick rapid fire round of questions.
Sure.
Just a couple of series of questions. I just want you to give quick, short answers to each of them.
All right.
Okay, so what do you think is one of the biggest unanswered questions in computational neuroscience right now?
Why the brain has so many cell classes, so many different kinds of neurons? Why?
What technologies are the most exciting to you right now?
Right now, for me, it's looking at voltage sensitive dyes and activators. So being able to image individual neurons at the membrane potential level. This is exciting.
And if somebody else was trying to enter this field, if a student was trying to enter this field, what advice would you give to them?
Linear algebra. Linear algebra is that's people I find don't walk into neuroscience knowing linear algebra. They come in knowing a lot about calculus. Calculus is great, but nobody's going to get you to do some, spherical integral. But linear algebra is the tool that we use to understand dimension.
Right. Okay. And what do you think is a mindset that would help people succeed in research in general?
So in general, sort of, be focused on a problem, right, and be able to sort of for a brief period of time focus in on something and ignore other aspects. That's how you kind of get deep into something. And when you're done that problem, then step away and ask what's the next problem, and it's okay to pivot in those scenarios. So there's periods of time where you have to be singularly focused, and there's periods of time where you have to take your head out of the sand and look at the world and say, what's the next thing I want to do? And having that flip-flop between those very different ways of looking at the world is important.
Right. Okay. So last question. If somebody listening could only remember one thing from this episode, what do you want that to be?
That understanding how the brain works requires you to record from subsets of the nervous system And that's enough, because the way we think computation is happening lives in low dimensional spaces, because computation is about coordination between activity. And it's not just an ideal gas model of the neuron, of the brain, where individual neurons are just doing whatever they want and they're independent of one another. No, it's the coordinations that make computation possible. And the signature of that coordination is lower dimensional activity than is possible to have.
Right. Yeah, there's definitely lots of synchrony and coordination that people ignore often.
And that coordination is low D. The signature of that is lower dimensions than is possible to have, and that makes doing my job a lot easier. I love low dimensional things because I can understand them. High dimensional things is very hard.
Yeah, for sure. Thank you so much for joining me today and helping really just make this complex technical field feel so approachable and exciting.
Great. Well, happy to do that. Thanks so much for having me. It's a lot of fun.
Yeah, of course. Computational neuroscience isn't just biological, it's really dynamic and interactive. I think we really found that out from today's episode. For listeners, if this episode changed the way that you think about the brain or sparked curiosity about neuroscience, AI, or just computational in general, share it with somebody you think would enjoy it. If you guys enjoyed this episode and learned something new, make sure to follow on your favorite podcasting platform or on YouTube. I'm Avnish, this is The Early Perspective, and I'll see you in the next episode.